So in the yesterday class, we discussed about what are sample, the monochromators, right? So in the monochromators, we saw the monochromators which are prepared through by using a prism, and we also discussed about the wavelength selectors like diffraction grating and all. And then we saw which is important and which is having more applicability in the instruments. That's what we saw. Then we discussed about sample containers. So the quartz gels, fused silica gels are used as a common sample holders for the UV visual spectrophotometry. And we also discussed about solvents which we can use. So we have to select the solvents only which are which cannot interfere with the absorption of light, especially at the specific wavelength which is meant for the molecule, solute molecule which we want to characterize. So that we discussed in the last class. And detection devices we have seen. So what are the various detection devices like photocells, phototubes, and photomultipliers? So they have their own importance in characterizing the absorption of a particular compound. So that's what we discussed in the last class. Okay, so the next one is called as amplification and readout devices. So the amplification devices will be always there only as and when they are, so as and when they are producing very little amount of energy. So if that amount of energy is not readed by the readout device, that means recorder, and then we'll go for the amplification. So that's what we saw in the photo multipliers. What happens when it produces a low amount of light, but it can be, so the low amount of light falls on the photocathode, but it releases only one or two electrons which may not generate electricity. So that's why we use diodes intervening, like cathodes, you can say intervening cathodes, and then automatically what happens, we can see there is an amplification in the number of electrons, and finally you can see there is a lot of change or lot of generation of electricity, and that electricity is directly proportional to the amount of electrons generated at the initial stage when the light falls on the photocathode. So such a way that we can calculate the amount of electricity generated by this particular light. Okay, so the amplification device, sometimes what happens when we have like phototubes and photocells and phototubes, and during that time, if the compound is absorbing very less amount of light, and then we can't able to record it in the recorder, and then automatically what happens, we have to be very sure that the recorder should be uh, the recorder should be recorded. So for that, what happens, we keep an amplification device. Amplification device will increase, amplifies. See, for example, there are two protons are released, and then these two protons will become into four, four into six, six into eight, or like exponential way, like 2, 4, 8, then 16, 32, 64, something like that. So in such a way that what happens, you can see there is an increase in the photon energy, and that photon energy will be measured. So the increase is mediated by the amplifiers. So the amplification will be done for the initial emitted light intensity will be get amplified, and then finally it can be calculated. So you know what, how many times you are amplifying. So based on that, you can easily calculate what is the initial intensity or what is the initial concentration of a compound. So that we will calculate. And readout devices are the devices, you can see something like the electronic signals can be read either in the amplifiers, or you can say like ammeters. That means, if for example, if you are measuring the electricity, and definitely you'll go for the how we can measure the electricity. Electricity can be measured by using the ammeters, right? ammeters or potentiometer or even potentiometric recorders and so on. If you maybe if you saw some of the electricity people, or you can like uh, helpers or something like that. Okay, so what happens, they will have a readout device. So when they touch with the wire, and then it shows what is the amount of electricity passing in that particular circuit. Okay, so similarly what happens, we will also use the same kind of meters like ammeters. Ammeter can give you the reading will be shown. So likewise what happens, you can see what is the amount of electricity generated by the photons, so that we can calculate. And potentiometers, potentiometric potentiometers and potentiometric recorders can also be used for characterizing or measuring the electricity, because electricity is electric signal generated is directly proportional to the transmitted light in this case. Okay, so these are all about the instrumentation. So in the instrumentation, you can see the instrument starts with the light source. So we know what kind of light sources can be used, right? So for UV visible UV light, you have to use some single light or something like UV light is always something like what we can say xenon lamps can be used, right? Xenon lamp can be used, and whereas a hydrogen lamp or deuterium lamp can be used for the visible light. So likewise, there are two types of lights which can be used in the UV visible spectrophotometer. So the light source, and the next one is a monochromator, or you can say that like um, the one which produces which converts the polychromatic light into the monochromatic light. So that also we will see. Okay, so the monochromators are again of two types here. So the monochromators will have three types, you can say, which we can collectively called as wavelength selectors, right? So wavelength will be selected by using this instrument. So that's why wavelength selectors are of three types. First one is filters, and filters can be seen only in the colorimeters, and the colorimeters are very specific for the to show the bandwidth but not the specific wavelength. Okay, so there are different filters which I have shown in the previous class. There you can see there are different colors, and then which are essentially required for the selection of a bandwidth of a particular ray. Okay, so maximum bandwidth is like 40 micrometers, or you can say that nanometers. Okay, this is what we will see. And the next is uh, in the wavelength selectors, the next one is monochromator. So the monochromator will be prepared with the in a closed box, so where we can see the light will pass through a entrance slit, and which falls on the collimating device, and collimating device will direct the light towards the reflect the light towards to fall on a prism, and the prism will have a um, diffraction grating. So diffraction will takes up, and so that the light will be separated into its individual components, so that means individual wavelengths. And then again what happens, the light which is coming out will be fall on the another lens, and then it directs the light, the single monochromatic light to escape from the monochromator box. Okay, so the exit slit will be there, and then that particular wavelength will be escaped from that particular exit slit. Okay, so then afterwards, and you can also see that there is another wavelength selector that is called as diffraction grating. So instead of prism, the diffraction grating will be there, and the diffraction grating will also is a um, uh, accurate one. So that's why you can see in almost all the modern instruments are holding the diffraction grating as the monochromator box. Okay, then the light monochromatic light which is coming out from the monochromator so will be passed through the sample. So the samples will be placed in a sample holder, which we call it as cuvettes, and there are different types of cuvettes are there, like you can use evacuated glass tubes as um, sample holders for the gases. And whereas for the UV light, so UV light can also absorbed by the glass, so that's why we can't use the glass for the UV range. Whereas for the visible region, you can use normal glass. But when we talk about UV visible spectrophotometry, so we need only one kind of sample holder. So that's why we use quartz quartz or silica quartz cuvettes, and which will not absorb either visible light or by the UV light. So that's why we use combinedly for all the samples we use. It may be the characterization at the visible region or at the UV region. So we will place the sample in the cuvettes. Okay, the cuvette is a quartz cuvette. Okay, where there is no interference. And then once the light is absorbed and then emitted and so on, and the emitted light intensity will be measured. So the measuring of emitted light intensity is always done with uh, placing a instrument called as um, what we call it as like um, detecting devices, detectors, we can say detectors. So they detect the amount of light which is generated or emitted from the sample. After the interaction with the sample molecules, the incident light will become into emitted light. So the emitted light intensity will be measured by using the detector system. So there are three kinds of detector systems available, and the three kinds of detector systems are like, the first one is detector system, uh, which we call it as like photocells. So the photocells are selenium based ones, and then so you can see that the first type of detector system which is identified when the photo spectrophotometers were identified. So that's why the photo cells has very importance in the earlier days. But it has given a base that how we can calculate the amount of light absorbed by the sample. Okay, so the optical density can be measured. So photo cells will be used, and photo tubes are there again. Photo cells, photo tubes, and photo tubes are again evacuated glass tubes with a semi-cylindrical uh, cathode. Semi-cylindrical cathode, inside of semi-cylindrical cathode is coated with the, it is coated with alkali and alkaline earth metals, and there is a centrally located anode is there. And the when the light falls on the semi-cylindrical cathode, inside of the semi-cylindrical cathode, there will be a, so there is a some kind of application of the alkali and alkaline earth metals in the inner side of the semi-cylindrical cathode. So when the light falls on the semi-cylindrical cathode in inner side, then these alkali and alkaline earth metals will react and they receive the photon energy, and then they release electrons, and the released electrons are collected by the anode and then generates the electricity which is connected to the circuit. Okay, so the amount of electricity generated is directly proportional to the amount of photon energy received by the photo cathode, or you can say that the alkaline earth metal cathode. Okay, so similarly, sometimes what happens, there may be a less amount of photon energy released from the molecule. So in that case, the readout devices may not be that much sensitive. They may not recognize that low levels of electricity which is generated. So in that case, we use another kind of the detector that is called as photo multiplier. So the photo multiplier can multiplies the incident electron circuit, and so that it increases the number of electrons every time. So the diodes will be there in the intervening the two electrodes, that is cathode and anode is intervened by the diodes, by placing diodes, and the diodes will amplify the potential, and then they move to the low potential areas. So such a way that what happens, they will move like this. So finally they reach the anode. So when they reach the anode, and then anode is connected to the circuit, and so you will be knowing what is the amount of electricity generated from these molecules. Okay, so from this absorbed from light. Okay, so that is directly proportional to the, that one is directly proportional to the amount of photon energy released from the sample. Okay, then we call it as like optical density, we can call it as like even transmittancy, you can call it as whatever it is, but you can measure that. Then the last one is amplification and readout device, and the readout devices amplification will be done only when there is a low amount of light is generated, which is not recognized by the detection devices. And so that's why we place the amplification device, and the amplification device will amplify it like um, that is photo multipliers. Like photo multipliers, you can see that. Okay. So then, uh, whereas in case of readout devices, usually the readout devices are the electric devices which can show what is the amount of electricity generated in a particular time. So likewise, what happens, you can calculate what is the amount of electricity generated. So as and when there is a application of the sample. So that's what we can see. And the readout devices also include like printers, because you can straight away you can link the system and then you can take the printout. So we may get the graphs, right? So that graphs can also appear in a modern spectrophotometric instruments, and those graphs can also be drawn with the readout device. We can see that in the screen, but we cannot download it, but you can get it printed so that you can have the hard copy. So that also a readout device. Okay, so the amplification and readout devices we already saw, right? Okay, so the next one is applications of the spectrophotometer, UV visible spectrophotometer. So what will happen with the UV visible spectrophotometer then? So UV visible spectrophotometer is can be used for various purposes that includes like qualitative analysis, quantitative analysis, enzymatic analysis, and even molecular weight determination, and then study of cis trans isomerisms, and even other physicochemical character studies. So all these can be characterized by using the UV visible spectrophotometry. I'll explain you one by one here. So the first one is qualitative analysis. How we can use the spectrophotometer in the qualitative analysis. So the UV visible spectrophotometer may be used to identify the classes of compounds in both the poor state, pure state, and in biological preparations. That means you can isolate samples from soil. Okay, so and then you can subject that for the characterization of the compound. So you are not extracting these compounds from the biological systems, I'm saying. Let's imagine if you want to calculate what is the amount of calcium or copper sulfate. What is the color of copper sulfate? What is the color of copper sulfate? Anybody knows? So we can do the qualitative analysis of molecules, right? So in the qualitative analysis, so we'll go for the spectra to identify the classes of compounds, whether they are in the pure state or in the biological preparations. So you should understand here that the pure state in the sense, so uh, chemical molecules, like you can isolate some of the compounds from like ores, right? So like, see, if you isolate the iron, so you have to calculate the iron concentration. Then in that case, what happens? We use the spectrophotometer. And whereas in biological samples also you can characterize the molecules. So in biological samples, especially, you can so the macromolecules are like proteins, carbohydrates, amino acids, then fats and nucleic acids. So all these things we can characterize by using the spectrophotometer. Okay, so the spectrophotometer will help us to identify the compounds either in the pure state, that means the natural compounds, or in the from the biological preparation. So these two kinds of molecules can be characterized. So that's why the chemistry people, they use spectrophotometers, and the biological people will also use, biological researchers will also use the spectrophotometers. Okay, so the range of absorption usually, so it determines what kind of compound will be there. See, because here I am saying that the qualitative analysis in the sense, we can identify what type of compound present in the sample, not the quantitative analysis. We have quantitative analysis also that we'll see in the next slide. But in case of qualitative analysis, you can assume what kind of compound is present in the sample. That's what how we can assume that. So it's all depends on in what range the compound is absorbing light. So that means we'll go for the complete range of radiation. Okay, range of radiation, which gives us, right, which gives us a good indication that from here to here, and then you'll be knowing where exactly it has the peak and all, right? So you can see the characterization or the behavior of the compound at a particular wavelength or given wavelengths, a range of wavelengths. So in that case, you can characterize what kind of molecules will be execute the absorption at a particular wavelength. So let's see the table here. See the absorption range and the structure and type of the compounds which we can identify here. See, for example, if the compound absorbs 220 nanometers to 280 nanometers range, so that means the absorption of light of a particular compound is done at 220 nanometers to 280 nanometers. So in that case, you can say these compounds may be, the compounds which are present in this may be aliphatic or alicyclic hydrocarbons. You know what is aliphatic and alicyclic hydrocarbons, right? Are their derivatives. You know what is aliphatic? You know or not? Anybody knows or not? Aliphatic hydrocarbons. Can you give me some examples for aliphatic hydrocarbons? So you have studied organic chemistry at your BSc level, even at the intermediate level, right? So if it is the case, you must know what is aliphatic and what is aromatic. Can you give me some example for aromatic aromatic compounds, hydrocarbons, aromatic hydrocarbons? Anybody?
సార్ అలిఫటిక్ అంటే చైన్ ఫామ్ అవుతుంది సార్.
చైన్ ఫామ్ అవుతుంది.
ఆటోమేటిక్ అంటే రింగ్ ఫామ్ అవుతుంది సార్.
రింగ్, రైట్.
రింగ్ లైక్ స్ట్రక్చర్స్ ఫామ్ అవుతుంది.
Okay, very good. Okay, so the aromatic compounds are the compounds which shows general normal linkage of the carbon atoms, like alkenes, alkynes, or even like cyclic alkenes, and so on. Okay, so you can see that they are not formed as a ring, whereas aromatic compounds are the compounds which will show a ring like structure with the unsaturated bonds. Saturated bonds will be there and rings will be formed even in the aliphatic compounds, but in aromatic compounds they are always having either double bond or triple bond, and mostly the double bond will be there in their ring. Okay, so the conjugative double bonds, the compounds which shows the conjugative double bonds with a ring, is called as aromatic hydrocarbons. Okay, so the aliphatic hydrocarbons will show the characteristic absorption at 220 nanometers to 280 nanometers. This is very fixed one, and then based on that, you can say the sample is having aliphatic or alicyclic hydrocarbons or their derivatives. That's it. The next is the compounds which absorbs at 220 nanometers to 250 nanometers, 220 to 250 nanometers, and these compounds usually contain two unsaturated linkages. Two unsaturated linkages in conjugation also be due to the benzene derivative. So that means the aromatic compounds can be can shows a characteristic property of absorption at 220 to 250 nanometers. This is what you should know, and this is very specific that they will show only one conjugative double bond, one conjugative, two unsaturated linkages in the sense one conjugated double bonds. That means double bond, single bond, and double bond, right? So that is conjugated double bonds. And then 250 nanometers to 30 nanometers molecules absorption range is specific for the more than two conjugated double bonds containing molecules. So they absorb light at 250 nanometers to 330 nanometers. So if there is a range of, if there is a absorption range in between 250 to 350 nanometers, and then we can clearly say that these molecules are having more than two conjugated double bonds. Okay. And then next is 450 nanometers to 500 nanometers. And this 450 nanometers to 500 nanometers usually is characteristic absorption of beta carotene, a precursor of vitamin A, which has 11 double bonds in a conjugated system. So, and and the color of this compound, it appears as yellow in color. Okay, so this is what we will see with the beta carotene. So the beta carotene has a specific absorption range that is called as 450 nanometers to 500 nanometers. And then some compounds will also show the 250 nanometers to 330 nanometers, and there where you can see like 249 nanometers to 60 nanometers, 325 nanometers, and they may be having um, naphtha quinone rings will be there for the in that particular compounds. Okay, and that's why they show a characteristic absorption. So that means based on the absorbance region, so you can identify what kind of compound is present in the sample. That's what we can see with the qualitative analysis. Okay, and the next is a quantitative analysis. So in qualitative analysis, you can also characterize uh, differently. So you can get the graphs and all for all the compounds where they will show different kind of absorbance. You see, qualitative. Then quantitative analysis is another kind of analysis. So the quantitative analysis in the sense we can calculate the amount of compound present in the sample. In the previous one, we can identify whether the compound is present in the sample or not. But now we can say that what is the amount of compound present in the sample. So I'll give you best example here that the quantitative analysis of spectrophotometric method is always helpful in calculating what is the amount of protein present in the biological sample, what is the amount of DNA present in the biological sample, what is the amount of RNA present in the biological sample, or fats or carbohydrates or any other molecules that we will calculate with the quantitative analysis. And quantitative analysis is very specific that you see, uh, I can say the nucleic acids, they will show the characteristic absorption at 250. Actually it is 260 nanometers, not 254. You can say 254 to 260 nanometers, and whereas proteins at 280 nanometers. So these are the good examples that we can calculate the concentration of a particular uh, compound present in the sample. See, the absorbance at 280 nanometers by the proteins depends on their two kinds of amino acids that is called as tyrosine and tryptophan. So the tyrosine and tryptophan are specific amino acids or the aromatic amino acids which shows the characteristic absorption at 280 nanometers. Okay, so based on that you can calculate what is the amount of protein present in the sample. Okay, and where do we use this particular uh, quantitative methods of spectrophotometric methods? So usually we use the estimation of proteins by Lowry method, estimation of tyrosine by Folin phenol method. We can call it as Folin phenol method, and then estimation of blood glucose levels by the Folin Wu method. So there are different methods which are there even for the DNA characterization or DNA concentration or RNA concentration also we'll go for these particular methods. So in these methods what happens, we finally will develop the color. So that means we can measure the sample either in the UV region or in the visible region. So you can develop the sample, you can develop the color so to read in the visible region, or you need not to develop the color if you want to read that in the UV region. Okay, so in case of proteins and in case of uh, nucleic acids, without any reaction, say for example, I'll give you an example here. So when we have a uh, for example, in the examination you have given a question that isolate and quantify the DNA from E. coli. So what will you do? So you'll go for a procedure that, so you will take the bacterial cells, and the bacterial cells will be lysed, and then we'll go for the extraction of DNA, and finally what happens, the precipitation will be precipitated, and precipitated DNA will be dissolved in the water or TE buffer or whatever it is. So once we have done that, then you have to quantify it. So what is the amount of DNA present in the cells, present in the sample? So for example, if you have taken like 5 ml of or 1 ml or 500 microliters of sample, bacterial cells, and you have to compare 500 microliters of the bacterial cells is consisting of this much of DNA. That's what you have to give the inference, right? Results. So for that, what happens? You have to calculate what is the amount of DNA present in that particular sample. To measure DNA, there is a standard formula for that. Like in case of double standard DNA, if you get one OD, so the one OD is equals to 50 micrograms of DNA. One OD is equals to 50 micrograms of DNA. Whereas in case of single standard RNA molecule, so that means in case of RNA molecule, one OD is equals to 40 micrograms of RNA. 40 micrograms of RNA. So if you know this standard formula, then let's imagine in the test you got something like 0.5 OD. So you are measuring the DNA concentration and you got 0.5 OD. So in the standard formula, 1 OD is equals to 50 micrograms of DNA. So now I got 0.5. Then what will be the concentration here? What will be the concentration? See, 1 OD is equals to 50 micrograms of DNA. And whereas 0.5 OD I got in the sample. Then what will be the concentration of my sample? Anybody? What is the concentration? You don't know. Tell me.
25, sir.
Yes, 25. It's very simple, right? So 1 OD is equals to 50 micrograms in the sense. So 0.5 OD is half of 1, right? So definitely it should be half of 50. So half of 50 is 25, right? So 25 micrograms is the correct answer. So such a way that we can calculate the amount of DNA, and there are different methods for the different procedures. Like Lowry method, we have a different method, and Phenol method, we have a different method, and glucose estimation, we have a different method. So those methods we incorporate, we'll get the optical density of a sample, and then we'll incorporate that in the formula, and then we can calculate what is the amount of compound present in the sample. So the quantitatively also you can calculate what is the amount of sample present in the sample, even amount of compound present in the sample, even in the biological samples, not only in the pure compounds. And you can also characterize, you can also quantify the biological samples. The next one, and the next one is enzymatic analysis. Okay, so as a biochemistry students, it is most important that you all need to know how we can characterize the enzymes, especially by using the spectrophotometric methods, how we can estimate the activity of the enzyme. So when we see the activity of an enzyme, so we have to be very sure that we are not doing any mistake in that. Let me give you an example here. See, in the last classes, I told you there is a special reference, special word called as blank or reference. A blank or reference is the one which is holding all the reagents except the one which is our interest, right? So that means what happens, we use that, and then we'll keep that, and we'll make that absorbance by these molecules as 0, if you remember, right? So we make it as 0, and then afterwards what happens, we use that as a reference solution. So as and when we place the another tube with the enzyme and all, what happens, enzyme will produce the product, and we want to characterize what is the amount of products which is produced in the reaction. So In that place, what happens? So we are going to measure only the product. But in case of any enzymatic reaction, you can see that it has substrate, coenzymes, cofactors, and other factors, and other all buffer and all interference will be there. But what we are doing is all these molecules absorbance will be made as zero, and only the newly forming product, that is called as a products, and product optical density will be measured, right? So similarly, in case of enzymatic analysis, we have to be very clean that always you have to link a specific kind of reaction to the enzymatic reaction. It may be the substrate or it may be the product, which should which concentration will determine the activity of the particular enzyme. Let's imagine I have a example here. So the lactate dehydrogenase is there. So the dehydrogenase is an enzyme which converts the lactate in reaction with the NAD plus. It gives rise to pyruvate and NADH plus H plus, right? So this is the reaction. So then what should I take? So I have like two substrate molecules here, right? One is lactate and NAD plus cofactor, and then pyruvate is the product, and then byproduct is the NADH plus H plus, or you can say like cofactor again. Okay, so then how can I characterize or estimate the activity of an enzyme in this reaction? So for this, LDH is lactate dehydrogenase. Lactate dehydrogenase is engaged in the transfer of electrons from lactate to NAD plus. Lactate to NAD plus. And the products of the reaction are pyruvate, NAD, and uh, a proton and a proton. One of the products is NADH, and which absorbs the radiation in the UV range at 340 nanometers. So what I want to see what compound will show the characteristic absorption at UV region or visible region. So visible region in the sense it is always linked to the intensity of the color. And whereas in UV region, you should always think what compound is essentially absorbs light at UV region. Okay, so in this reaction, only this NADH will absorb the light at 340 nanometers. At 340 nanometers. So I'm going to use NADH concentration to characterize the enzyme activity. So how can I do that? You see, and the reaction in the forward direction can followed by measuring the increment in the light absorption of the system at 540 nanometers in the spectrophotometers. Okay, so let me check. It is 340 nanometers, right? It's only at 340 nanometers, not 540 nanometers. It is 340 nanometers. Okay, so when I place a sample in a tube, right? So the sample in the sense, so it has lactate, then NAD, and then lactate dehydrogenase is added. All the buffers are there in the tube. Then now what happens as and when add the lactate dehydrogenase, then the reaction will start, right? So that means I'll place the sample in the in the cuvettes place, sample holder place. Then I'll get zero at the time of zero at the time of uh, initial reading. That means when there is no reaction is started, zero. Let me tell you why it is zero. So the solution is having lactate, NAD, and lactate dehydrogenase. Because I am saying that I have fixed at 340 nanometers, the machine is fixed. So then it measures only the optical density at that particular wavelength. So now, as and when I add these two, yes, Emma, any problem? So as and when I add lactate and NAD, because there is no NADH here, so that's why what happens at 340 nanometers, it shows zero optical density, zero OD. And then as and when I add lactate dehydrogenase to this reaction, then automatically lactate dehydrogenase mediates the conversion of lactate into pyruvate molecule. By the time what happens, it also converts the NAD plus into NADH plus H plus. So initially it is zero, and I'll keep it for like 20 seconds. So by 20 seconds, what happens gradually? There is an increase in the optical density because there is an increase in the NADH. Are you clear? So there is an increase in the NADH, then automatically there is increase in the optical density. So when the optical density is increases, so that means it shows that the reaction is in progressive manner. So it is progressing. Enzyme activity is going on, right? So likewise, I'll take the optical density for this reaction for something like 3 times or 4 times or even 5 times, 6 times. That means with an interval of 20 seconds. So the first 20 seconds at 0th time, it is 0. First to 20th seconds, there is one word is something like 0.2. And then next one, that means next to 20 seconds, that is 40 seconds. At 40 seconds, it is like 0.4. And next is 60 seconds, right? So that likewise, what happens, I will go for either 2 minutes or 1 minute. It depends on what you want to do. So that's why what happens, you can see there is a progress in the NADH. Okay, so that means the increase in the concentration of the NADH is directly proportional to the enzymatic reaction. Are you clear with this? Is any doubt for you in this case? Is it clear?
Yes, sir.
Yes,
Yes.
sir.
So it is clear. So all of you clear, right? If anybody is not, understood, you please let me know. I'll explain you once again. So in the enzymatic reaction, you can see that we select either products or the substrate molecules as a characteristic property at either UV or visible region. So in this reaction, that means lactate dehydrogenase mediated reaction, we select NADH as the characteristic molecule for characterizing the characteristic absorption at 340 nanometers. So because the reaction, it is the end product, so that's why what happens, you can see there is an increase in the NADH, and which is directly proportional to the enzymatic reactivity, or you can say that the enzyme activity. So that's why we will add, initially we will add lactate and NAD, we make it as 0, and then we will add lactate dehydrogenase. After 10 minutes, 10 seconds, what happens, you'll go for the reading. So you'll get some reading, because by that time there is a conversion of lactate plus NAD into pyruvate and NADH. Okay, so as and when the NADH in concentration increases, you can see that the absorption, or you can say that the optical density will also get increased. So when it increases, that is directly proportional to the rate of reaction of the enzyme. Okay, so this is what we will do. Okay, so when a reaction is having such a kind of molecule, and then we can characterize it. If I have a reaction and it has like substrate molecules and the product molecules, but all of them will not show any characteristic absorption at UV or visible region, then what I have to do? Do you understand what I mean? So when we have the direct reaction, and any one of the product or the substrate molecule is having the characteristic absorption, characterize it. If a reaction which doesn't have any characteristic property of absorption, characteristic of absorption at UV or visible region, then what I have to do? I want to characterize it also. I want to see the enzymatic activity of this reaction also. In that case, what we will do is we will link up the reactions, join the reactions, and the reaction which has the characteristic absorption at 340, at UV or visible region will be linked to that reaction, and then we can characterize the or then we can characterize the enzymatic activity. Let me give you some example here. So we already know that the the lactate dehydrogenase can convert the lactate molecule into lactate NAD plus into pyruvate plus NADH plus H plus. So now this one is having NADH is having 340 a characteristic absorption at 340 nanometers, and we characterized it. But now what happens? I'm going for the next reaction. So the next reaction where I can see the pyruvate kinase enzyme, pyruvate kinase, it converts the phosphoenol pyruvate plus ADP molecule into pyruvate and ATP molecule. It converts the phosphoenol pyruvate plus ADP molecule into pyruvate and ATP molecule. I want to know what is the the enzymatic activity of pyruvate kinase for this reaction. And you see the phosphoenol pyruvate plus ADP or ATP, all these four molecules will not show any characteristic absorption at UV or visible region, right? And the pyruvate, so this reaction is always linked to the another reaction, and so such a way that what happens, you can calculate the amount of uh, light absorbed. Okay, so let me explain you how it happens. So phosphoenol pyruvate plus ADP, what happens? It gives to the pyruvate plus ATP. And you see the next reaction for pyruvate is, what is that? It is nothing but the lactate will be converted. Lactate, when lactate reacts with the NAD, then what happens? What happens? It gives rise to pyruvate and NADH. Is it not? So that means it is already, the pyruvate is the substrate for the conversion of pyruvate plus NADH plus H plus into lactate plus NAD plus. See, in the previous reaction, saw that the lactate dehydrogenase enzyme, this is a reversible reaction. Lactate plus when reacts with the NAD plus, which gives the pyruvate and NADH plus H plus. What we saw in the this one. And whereas we are reversely connecting this reaction to the another reaction, so that we can characterize, we can know what is the activity of pyruvate kinase in converting the pyruvate molecule. So that means, you see, the phosphoenol pyruvate plus ADP gives rise to pyruvate plus ATP, and we use pyruvate, and then we will add NADH plus H plus to this reaction, and we will add the lactate dehydrogenase enzyme, and this lactate dehydrogenase enzyme will convert the pyruvate plus NADH plus H plus into lactate plus NAD plus molecule. What happened here, you see, we are adding to the reaction this NADH plus H plus, and the characteristic molecule which absorbs the UV region or the visible region is the NADH plus NADH, which absorbs at 340 nanometers. I'm going to add NADH to the reaction. That means I know how many moles of NADH was added to the reaction. When I know the moles of reaction, then I will be knowing. So when I place that tube initially at the zero time, without the addition of the lactate dehydrogenase enzyme, right? So then automatically what happens? I have pyruvate, which is produced from the phosphoenol pyruvate, produced from the pyruvate kinase activity, and I am adding the NADH, and NADH is giving some OD, something like it has like 0.1.5 OD. So I am going to add some moles of the NADH, and that is giving 1.5 OD. Imagine. So at the initial stage, it has 1.5 OD. And then later what happens, when the pyruvate molecules pyruvate is formed, then automatically the pyruvate and NADH will react together, and then it forms as lactate. So that means what happens, the NADH is getting depleted, and then it forms as NAD plus. Bhargavi, Bhargavi. Okay, so what happens here? The pyruvate reacts with the NADH, and then NADH will be converted into NAD plus. Let's imagine that there are 100 molecules of NADH when I added to it. Then the pyruvate molecules, as and when the pyruvate molecules are formed, and the pyruvate molecule, first molecule will react with that, and then it converts into lactate and NAD plus. Now only 99 NADH plus molecule NADH are there, right? And likewise, number of pyruvate molecules, that means 1, 2, 3, 4, 10, 50, 50 molecules of pyruvates are formed, and that 50 molecules of pyruvate will be get converted into lactate and NAD plus. What will happen in the reaction? So there is a 50 molecules of NADH is depleted from the reaction. So it is 1.5 when there is 100 molecules. So what happens when there is a 50% of the NADH is getting reduced, then automatically there is a decrease in the optical density. So in this reaction, the previous reaction, we started with the 0, and every 10 seconds we are getting the reading, increasing the optical density. Whereas in this case, we have the maximum optical density at 0th point, that is 1.5. And then as and when the NADH is converted into NAD plus, and there is a depletion of NADH, and then automatically the absorption capacity will get decreased. That means number of molecules are not there. So that's why there's 1.5 will become into, after 10 seconds it will become into 1.3, then 1.1, 0.9, 0.7, 0.8, or 0.6, 0.5, something like that. So there is a decrease in the optical density, we'll see. So if the absorbing material or absorbing compound is present in substrates, absorbing molecule in the substrates, and then you have to take, you have to see the decrease in the concentration. If it is in the products, and you have to see the increase in the concentration. Are you clear with this? Is there any doubts? Emma,
No, sir.
do you no?
Hmm.
No doubts, or anybody want any clarification?
No, sir. No doubts.
No doubt. Farhana, no doubt.
No, sir.
Okay. Okay. So this kind of reactions can be called as coupled assays, because we are connecting one reaction with the another reaction, and then we are characterizing the enzyme activity. So that's why they can be called as coupled assays, right? Coupled assays. And there are so many reactions like that, like assay of urease enzyme activity can be done like this. Assay of celery amylase activity can be done, and effect of temperature on the uh, amylase activity can also be done. So likewise, there are several reactions which can be coupled and then can be characterized their enzymatic activities. Okay, so this is what the enzymatic activity that we can measure with the spectrophotometer. You can also measure the molecular weight of the compounds. Okay, so the molecular weight of the compounds, so usually, so we can calculate by using a special formula that is called as M is equals to AWB by OD is the one which we can use the formula. And there you can see the A is the extinction coefficient, and W is the weight of the compound, and B is the path length. So by using the standard value, so you can also calculate the molecular weight of a compound. Molecular weight of a compound. So for example, molecular weight of amine picrates, sugars, and many aldehydes, ketone compounds, and uh, and other compounds are also determined by using the UV visible spectrophotometry for their molecular weights. So for molecular weights, there is a specific formula that is called as capital M is equals to a small a W small b, small w, small wb by OD. So in this case, you see A is a extinction coefficient, W is weight of the compound, how many grams you have taken, how many grams are dissolved in liter or half liter or whatever it is. So that is the concentration, weight of the compound. And then the path length is always fixed with the cuvette. So it may be 1 centimeter or 0.5 centimeters or even 0.1 centimeter, it depends on. Okay, so these three kinds, and then we can calculate the molecular weight of the compound. And besides that, you can also, and this molecular weight determination is very specific. It can be uh, specific to the only small molecules, not for the large molecules. So the small molecules can be subjected for the molecular weight determination in the UV visible spectrophotometry. The next is study of cis trans isomerism. You know what is cis and what is trans isomerism. If you remember, in case of glucose, especially in chemistry, you maybe studied what is cis forms and trans forms, right? Cis and trans isomerisms. Is it not? Emma, have you studied the cis and trans isomerisms? I need answer.
Yes, sir.
Yes, sir.
Okay, so you know what is cis and what is trans isomerisms. Okay, so usually the cis and trans isomerisms can be measured. So usually how it forms is it is a geometrical isomers. Geometrical isomers in the sense, if for example the compound forms like this. This is one form. Sometimes what happens because of some relaxation, so it forms like this also. That means there is a change in the structural arrangement of the compound. So that means like this and like this, right? So that means it automatically indicates that this is one form and this is another form. Okay, so which we can call it as, like, one is called as cis and another one is called as transform. And let me tell you what is cis and what is trans, and how we can identify them. So the geometrical isomers, they differ in the spatial arrangement of groups about a plane. The absorption spectra of the isomers are also different, because when there is a change in the arrangement, see, when it forms like this, when it forms like this, there is a change in the, you know, arrangement of the isomers. That's why the absorption will also get changed. And let's see, the trans isomers are usually elongated. Trans ones are trans trans isomers are always elongated ones, so longer ones. And whereas cis isomers are like somewhat shorter ones. Okay, so when there is a longer one, then the trans isomers have high wavelength of absorption, and whereas cis isomers are having less. See, because when it is elongated, then it has more surface to absorb, right? So that's why it absorbs more. And whereas when it is a smaller one, then automatically the surface absorption surface will get reduced. So that's why the absorption will be low. So you can say that if a compound showing high amount of absorption, maximum absorption at a higher wavelength, and then that can be called as a trans isomer. And the other one which shows in the counterpart, that means in the opposite way, that can be called as cis isomer. So do you understand how we can characterize it? So the elongated ones are the trans isomers, and the shorter ones are the cis isomers. And elongated ones will absorb more light, and whereas shorter or a low level of absorption will be seen with the cis isomers. Okay, so this is what we'll see with the study of cis and trans isomerism with the compounds. And you know that the compound, single compound, will show the both the forms, cis and trans. For example, take glucose. Glucose will have cis isomerism and trans isomerism also, right? So there is no change in the, number of elements present in the compound, but only the spatial arrangement will be different. That's what. The next is, uh, you can also use the control of purification. So impurities can be detected uh, from the samples. So for example, carbon disulfide impurity can be um isolated. Impurity in carbon tetrachloride can also be detected easily by measuring the absorbance as 380 nanometers. And so that what happens, we can easily identify what are the different kinds of impurities present in the sample, and then you can go for the purification and so on. So impurities can be identified. And besides that, this UV-visible spectrophotometry can also be used to identify the a large number of physicochemical characteristics, physicochemical studies. The physicochemical studies are the studies spectrophotometry has been used to study the chemicals. Especially, it is used in the heats of formation, molecular addition, compound and complexes in solution. Heat heat formation in the molecular addition, compounds and complexes in the solution. So when we add some compounds and then heat generates, that we will characterize. Then determination of empirical formula. You can determine the empirical formula. You don't know the compound, and then you can determine how many carbons are there, how many hydrogens are there, how many oxygens are there. Then you can compare that, and then you can calculate you can write the empirical formula. See, for example, the empirical formula for glucose is C6H12O6. Okay, so similarly for any unknown compound, you can identify the empirical formula. Then formation constants of complexes in solution. So the formation constants in the sense, for example, if you add two mixtures, and then what happens, they will form as a complex, and that complexes can also be identified and then can be characterized. And then hydration equilibrium of carbonyl compounds. So carbonyl compounds will be equilibrated with the hydrogen, so that equilibrium can also be calculated by using this. And association constants of weak acids, bases in organic solvents, that also we can characterize. And protein dye interactions, you can see how the protein dye getting interacted, and how they can be shown a characteristic property, that also we can identify. Then chlorophyll protein complexes can be studied, how they can interact one to other, and that we can characterize. Vitamin A aldehyde protein complexes, how it forms and how it helps in the biological reaction, that we can identify. And determination of reaction rates. So I already told you determination of reaction rates. In case of enzymatic reactions, you can also use the same kind of experiments for the um, uh, pure compounds and their reaction rates. And then dissociation constants of the acids can be identified. Association of the cyanine dyes can also be characterized. Okay, so these are the different physicochemical studies, physiochemical studies, which has been subjected to the characterization by using UV visible spectrophotometers. Okay, so this is what about the spectrophotometer. Is there any doubts? You can ask me now. Any doubts? Any clarification required? Yes, ma.
No, sir.
So you don't have any doubt?
No, sir.
Sir,
No. Is anybody having?
No, sir.
No. That's great. I don't know whether all of you is clearly understood, or all of you are not at all understood. There should be no when you don't understand completely. There should be no when you understand completely. There is in the middle, and definitely you'll have some questions. Okay, so that is up to you. If you don't want to ask me any question, then it's okay. Okay, then. So if you don't have any doubts, and then I leave the class, and from the next class we'll go for the infrared spectroscopy. Okay,
Yes, sir.
any doubts? Why Bashir is not asking any question? Bashir, is it not Bashir? Is Bashir in the class?
We don't know, sir.
I'm just looking at. Why this Guru Reddaya and Maunika is not added to the class? Are they in the class?
No, sir.
Ah, yes, is it Guru Reddaya?
Yes, sir.
Okay, so you are in the class?
Yes, sir.
Why many of you are as guests? You are guest. So guest ఉన్నారు కదా, గెస్ట్ ఉండే వాళ్ళందరూ హోప్ఫుల్లీ మేబీ యూ డోంట్ హ్యావ్ ప్రాపర్ రిజిస్ట్రేషన్ సరిగ్గా చేసుకోలేదు మీరు. ఒకసారి మళ్ళీ ట్రై చేస్తారేమో చూడండి రిజిస్ట్రేషన్. ఓకే.
ఓకే సార్.